Thank you for joining today's webinar. Today's topic is IVDR performance evaluation of in vitro diagnostics, key insights and legacy device considerations. Today we are excited to have Clemence and Jordan. Both are consultants at the Strategical, a product-like group entity. And basically, a quick reminder: this session will be recorded, okay? And we are happy to share this with you after the webinar. And also, just to remind you, there will be a Q&A session at the end of the presentation. So feel free to post your question in the Q&A section, and we will make sure to pass them to our speakers. I will read them and they will answer them. So Clemence, Jordan, if you want to start, feel free.
Thank you.
Yes, thank you, Fernando, for the introduction. So this webinar will be in three parts. First, an introduction to the notion of performance evaluation. The second part will develop how to demonstrate the performances of an IVD. And the final part will focus on legacy device and what can be reused from the directive. Okay, so let's start with the introduction to performance evaluation. Okay, so we all know that before an IVD can be placed on the market, the manufacturer must affix the CE mark to its device. And to do so, they must demonstrate compliance with the requirements of the IVDR. In particular, he must provide clinical evidence that the device is safe and achieves the expected clinical benefit with reference to the state-of-the-art in medicine. In other words, in addition to demonstrating the safety of the product, the manufacturer must scientifically demonstrate the positive effects of the device due to its function, such as screening, monitoring, diagnosing, or assisting in the diagnosis of patients. Or it can be the positive effect on patient management in terms of public health. This is a definition of clinical benefit in the IVDR. Now, if you refer to the definition of clinical evidence given in Article 2.36, it is the clinical data and the results of the performance evaluation which make it possible to assess whether the device is safe and whether it offers expected clinical benefits when used in accordance with the manufacturer's intended purpose. The performance evaluation is therefore an essential element in obtaining CE marking for an IVD. So, okay, I need to realize the performance evaluation of my product, but what it is, how it works. Article 2 gives a first definition. It is assessment and analysis of data to establish or verify the scientific validity, the analytical performance, and, where applicable, the clinical performance of a device. So we understand that there are several components. Then Article 5 precises that it is necessary to demonstrate the conformity with general safety and performance requirements. Finally, Article 56 says that it provides sufficient clinical evidence. So in short, to demonstrate that a product is safe and produces the expected clinical benefits, the manufacturer must provide clinical evidence. This clinical evidence is based on the three pillars of performance evaluation: scientific validity, analytical performance, and clinical performance. And to understand the importance of these three pillars of performance evaluation, we need to go back to the definition of an in vitro diagnostic medical device. So what is an IVD? How does it work? It can be defined by its function, such as screening, diagnosis, monitoring, prognosis, etc. The method it uses, what is the type of technology, whether it's automated or manual. The type of data it produces, is it qualitative, quantitative, semi-quantitative? what type of users it's intended for. Is it the patient himself, a healthcare professional, a trained personnel? And in what environment will the test be performed? It can be an analysis lab, at patient's hospital bed, at home, maybe in an ambulance, whatever. The input data for this device are the target population from which the sample will be collected, the sample in which the analyte will be measured, detected, or quantified. It can be blood, serum, cells, urine. The analyte itself, of course, it can be biomarker, chemical component, and enzymatic activity. In the case of companion diagnostic, the associated drug will also be taken into account, of course. And as output data, the analysis by the device will produce information concerning a clinical condition or a physiological state. So let's make a focus on the three pillars of performance evaluation. Now that the product is clearly defined, let's return to the three pillars supporting clinical evidence and make the link with the various components of an IVD. The first pillar corresponds to scientific validity. This is a new feature of the regulation which did not exist under the directive. Scientific validity is defined as the association of an analyte with a clinical condition or physiological state. It demonstrates the value of measuring, detecting, analyzing the analyte in question or its properties. And scientific validity is therefore the process of establishing a correlation between the analyte and the clinical objective of the IVD. In a way, scientific validity demonstrates how the device can have a positive impact on the patient's health. It defines the expected clinical benefits that the medical device must achieve and which the clinical evidence must demonstrate. In this picture, we can see that the data to be taken into account for the demonstration of the scientific validity are the target population, the type of sample, the analyte, and the clinical condition or physiological state concerned. Scientific validity will only use these elements, whatever the methods, the environment, the type of data, the users, etc. And we can see that scientific validity can be demonstrated independently of the IVD. Okay, Clemence, it's for you.
So we saw that parameters of an IVD are what parameters of IVD are involved for the scientific validity. Let's do the same work for the second pillar, analytical performance. The analytical performance of a device represents its ability to correctly detect or measure a given analyte. The analytical performances are established by considering one or more sample types, the analyte, method used and the type of data generated, users, environment, and the clinical or physiological condition are not considered for analytical performance demonstration.
Okay, and finally, after the second pillar comes the third, the clinical performance. The third pillar of performance evaluation, the clinical performance, is the ability of a device to produce results correlated with a given clinical condition or physiological process or state, depending on the target population and the user for whom the device is intended. The IVD components mobilized to demonstrate this clinical performance are different from those mobilized to demonstrate analytical performance, since the IVD's clinical performance will be defined by considering the target population, the device function, as well as the user and the environment in which the device is to be used. and the physiological state or clinical condition qualified by the device. In the case of companion diagnostics, the drug associated with the companion diagnostic will, of course, also be taken into account. So, to establish clinical evidence that the device is safe and offers the expected benefits, you understand that we cannot simply demonstrate scientific validity, analytical performance, or clinical performance alone, but it's the combination of all these three components that enable us to cover all the parameters of the device and provide the data needed to demonstrate the clinical evidence. Okay, let's make a first summary. We saw that performance evaluation is assessment and analysis of data to establish or verify the scientific validity, analytical performance, and where appropriate, clinical performance of a device. It allows compliance with general safety and performance requirements to be demonstrated, and it provides scientific evidence that the device is safe and offers expected clinical benefits. We will now discuss how to demonstrate different pillars of performance evaluation with this second part. First, the scientific validity. The demonstration of scientific validity is highly linked to the intended use of the device. To illustrate this point, let's take the example of 3 devices, all designed to measure C-reactive protein, the CRP, but with different uses. In the first row of the table, let's imagine that the claim of the first device is to determine the level of CRP in the serum of patient to assess the inflammatory state of the body, meaning to check that the level of CRP in the blood is within the normal range. To demonstrate the scientific validity of this kit, we will need to show how CRP protein plays an essential role in the body's inflammatory response and under what circumstances this level is abnormally high. Moving on the second line, if we now consider a device whose purpose is to diagnose or confirm a pathological condition associated with an abnormal level of CRP, in the case of rheumatoid arthritis, for example, we'll need to demonstrate that the abnormally high level of CRP in the blood is associated with rheumatoid arthritis. And the last example, let's imagine a third device whose claim would be to enable monitoring of CRP levels in patients treated with anti-inflammatory drugs to reduce the inflammatory state and hence the CRP levels. The manufacturer would have to provide proof that CRP levels are a good indicator of treatment efficacy, and that they enable the physician to adjust the drug dose. So as we can see, the evidence required to demonstrate the scientific validity of a device differs according to the manufacturer's claims for the device. The regulation specifies the sources to be used alone or in combination to demonstrate scientific validity. This will depend, of course, on the data available. The association of an analyte with a clinical condition or physiological state is often documented by academic research data or studies evaluating the potential of said analyte for a specific clinical application. Manufacturers can use this peer-reviewed scientific literature to provide evidence of scientific validity. Scientific literature means journal articles, conference posters, guidelines, or documents from official websites such as MedTech Europe, the WHO, the EMA, etc. The manufacturer may also use expert consensus opinions from relevant professional associations such as learned societies. If there are other devices measuring the same analyte for the same purpose, whatever the method used, the manufacturer may use the data available for these different devices to document its own scientific validity. And in the case of innovative analytes, for which scientific validity is emerging, it's likely that the manufacturer will need to demonstrate this through feasibility studies or clinical performance studies. We will take a closer look and detail when to use which sources of clinical evidence.
If there is a consensus, scientific validity is established. This is the case for devices such as pregnancy tests, where we know that beta-HCG is produced by the embryo shortly after conception, making it an ideal marker for detecting pregnancy. Another more recent example is PCR testing to detect people infected with COVID virus. The scientific validity of S or N protein RNA is widely described in the literature by the WHO and even in the guideline MDCG 2021-21. For these cases, there is no need for a systematic literature review. Reference to the guides or to a few reference documents in the field will be enough. The manufacturer can also use relevant information for other devices measuring the same analytic. But be careful, however, to quote references that correspond to the intended use of the device, the target population, etc. If there is no consensus to validate the link between the analyte and the physiological clinical state, a systematic search of the literature should be carried out to find evidence of the relevance of measuring this analyte for the intended use. Or identify other data, such as data on similar devices, design feasibility study, or other clinical utility study, for example. Once the data have been identified, their relevance to demonstrate the link between the analyte and the clinical condition need to be appraised and analyzed. By relevant, we mean data that demonstrate the link between the analyte and the clinical condition on the target population and corresponding to the device destination. When there is no evidence of scientific validity, or when such evidence is deemed insufficient or where the degree of novelty is high, new analytes or new intended uses, it will be necessary to provide scientific justification and generate new or additional data. The manufacturer can determine what additional evidence is required by carrying out a gap analysis. He may then specifically design a study to demonstrate scientific validity, or if clinical performance data are available, he may potentially reanalyze them to provide further evidence of scientific validity.
Okay. It's very important that the protocol and the method used to demonstrate scientific validity are clearly described by the manufacturer in the performance evaluation plan. When manufacturers carried out literature reviews or other data search methods, they must define the search strategy before starting the search. The strategy must be complete, meaning systematic, and objective. It must identify all relevant data, both favorable and unfavorable. Concerning the use of peer-reviewed scientific publications, the manufacturer should make an inventory of the sources used for the bibliographic research. What was the database, the internet website of plant societies consulted, the official medical recommendations, such as European and French websites, for example. He should explain the research strategy, the keywords used based on the name of the analyte, the medical indications, the target population. He should also explain the filters used to limit the query, for example, the publication period, the type of publication, etc. The idea is to focus the research according to the intended use of the device and try to make it as relevant as possible without ending up with too much data. The manufacturer should also explain how the information collected will be evaluated and analyzed to assess its relevance. The idea is to explain the criteria used to select relevant information based on its quality. And to be relevant, the source of information must refer to both the analyte and the clinical or physiological condition and show a clear link between the two. Information will be selected or rejected according to the appraisal, and the reason for exclusion will be detailed. If the search has been carried out using keywords in the abstract of the articles, it is possible that a selected article may turn out not to be very relevant after a complete reading. Maybe because the link between the analyte and the physiological condition is not so obvious, or the population to which the article refers does not correspond. In this case, you should keep the article reference, but explain why it was not selected. Similarly, you can use data from devices measuring the same analyte developed in-house or by other manufacturers. You will need to conduct a review of any internal or external documentation you consider relevant. And to do this, your search may be based on data available on Udemy, and in particular the summary of safety and performance, the SSP, of existing CE-marked products. You can also potentially use the data you will find in the instruction for use of other manufacturers' devices, or look at the patent data for these devices. In the same way as for bibliographic research, you'll need to explain the applied research strategy and then analyze the relevance of the information collected. We've talked about the first pillar of performance evaluation, scientific validity. Now let's look at how to demonstrate the device's analytical performance. Demonstrating analytical performance means demonstrating the performance of the method used by the device and must answer the question, does the device measure and/or detect the analyte reliably, accurately, and consistently? The relevant performance characteristics, which form part of the GSPRs and are linked to the analytical characteristics of the device, must be supported by existing evidence or by the generation of new evidence. In the vast majority of cases, this can be demonstrated through performance studies. The section 9.1 of Annex 1 in the IVDR, which I invite you to consult, lists analytical performances to be assessed by the manufacturer. In particular, the IVDR mentions analytical sensitivity, precision, accuracy, analytical specificity, measurement range, linearity, etc. These indicators may depend on the test technology and the intended environment of use. The importance and weighting of the various indicators listing in the section of the IVDR should be considered on a case-by-case basis, as not all are necessarily applicable. However, any omission must be clearly explained and justified. The choice of criteria to be evaluated in terms of analytical performance may be guided, among other things, by the results of the risk analysis, for example, measurement variability criteria to be taken into account, choice of interference to be tested, etc. Okay, let's come back to samples. If the manufacturer claims that the device can measure the analyte in urine or whole blood, analytical performance will have to be evaluated with these two types of sample. As another example, if the manufacturer claims that the device can measure the analyte from serum taken from both SST and dry tubes, the performance will have to be evaluated with both types of tubes. The manufacturer must demonstrate sample stability, taking into account the time between sample collection and analysis, storage condition and temperature limits, and must define the recommended number of freeze-thaw cycles. All this information must be provided and reflected in the analytical performance report. This is particularly relevant for IVDs using methods whose analysis must be completed within a given time frame. Let's turn now to clinical performance evaluation, which, as we've seen, involves demonstrating a device's ability to produce results that correlate with a particular clinical condition or physiological state in relation to the target population and intended user. The manufacturer should demonstrate that the IVD has been tested for the intended use, target population, use condition, operating and use environment, and with all the intended user group. Indicators of clinical performance vary and depend strongly on the intended purpose and performance claims. The IVDR sets out that clinical performance may not be required for certain devices in Article 2.39. For example, clinical performance data may not be expected for non-sterile specimen receptacles, microscopic glass slides, or some general reagents. In such cases, and where due justification is given, a clinical performance report would not be expected. The clinical performance of a device is demonstrated on the basis of one or more of the following sources. It can be clinical performance studies, peer-reviewed scientific literature, or published experience from routine diagnostic tests. And by published experience, we mean any document or dataset resulting from the use of the device that has been the subject of a brochure aimed at the general public, a publication on a laboratory website, an internal document available on request, an officially announced information, etc. Clinical performance studies are carried out unless it is duly justified to rely on other sources of clinical performance data. And as a second take-home message, we can see that the three pillars of performance evaluation, for example, scientific validity depends on the intended use. We saw that. We can demonstrate the scientific validity independently of the device, and the third strategy have to be well described in the performance evaluation plan. Analytical performances are generally the same for all IVD, but may vary depending on the function and the technology of the test. Finally, clinical performances depend on the intended use and the manufacturer's claim. Now let's focus on legacy devices. We've seen what is meant by performance evaluation and how to obtain clinical evidence that the device is safe and provides expected clinical benefits. We can ask whether obtaining clinical evidence is easier for a legacy device. When asked, is it easier to obtain clinical evidence for a legacy device? The answer is legitimately yes. Indeed, a legacy device is one for which analytical performance data, clinical performance data, design data such as data from feasibility study or risk management study are available. But also, potentially, data acquired after the product was marketed, such as PMS data, or data from medical economic studies or clinical studies carried out with the device by users of the device, etc. We therefore potentially have a mass of data which in theory should facilitate the scientific demonstration of the clinical benefit and safety of the IVD. In fact, as we can see from this diagram, the data at our disposal can, a priori, support scientific validity, analytical performance, and clinical performance, as well as compliance with GSPR. So we have quite a lot of data and at our disposal to provide scientific proof of the device benefits. But are all these data sufficient and can they be used as they stand?
There are certain points to consider before using previously collected data. As Jordan mentioned, scientific validity is a new concept introduced by IVDR. Although not formalized in the directive, this work may have been carried out by the manufacturer during the design phase. Indeed, it is hard to imagine to design a product without considering the link between the analyte measured and the information provided by the device. However, scientific validity is closely linked to the intended use of the device. Before using design or feasibility data, you need to make sure that the product's intended use has not changed. Moreover, the state of the art is constantly evolving, in addition to the evolution of knowledge and technological advances. The standard of reference tests on which you relied may also have changed. What was up to date a few years ago may no longer be up to date, so make sure your state of the art is accurate and up to date. Finally, IVDR has led to a tighten up the quality level of the data requested. If you are relying on literature data, this must be systematically evaluated and analyzed. Concerning analytical performance, the first question to ask is whether the current version of the device is the same as the one used in the performance studies. In other words, has the device undergone any change that might call into question the analytical performance data obtained previously? A change of supplier, a change of raw material, a change in the manufacturing process. Have all the characteristics of the device been evaluated? Interferences, type of sample, any change must be recorded and you must provide proof that these changes have no impact on the results previously obtained. If there is not the case, no new study will have to be carried out. Finally, with regard to clinical performance, the point of attention are broadly the same as those to be considered for demonstrating scientific validity and analytical performance. Is the version of the device used to perform the test the same as the one being evaluated? Changes in the manufacturing process, in the control use, in the reference material, etc. Are the intended use and claim are the same? In the case of a comparison study with reference tests, have you re-examined the state of the art concerning this reference test? Once again, any change must be recorded and you must provide the proof that these changes have no impact on the result previously obtained. If this is not the case, new study will also have to be carried out. So the question is it easier to obtain clinical evidence for a legacy device? The answer is yes, of course, but because you may have a lot of available data. But the application for CE marking under IVDR must be considered as an application for initial certification. It means that the use of the data available in the context of IVDR should not be considered systematic. You must ask yourself whether this information is relevant today for the evaluation of this version of the device. Thank you.
Thank you very much. Let's speak to Fernando to manage the Q&A phase.
Of course. Okay. Clemens, Jordan, thanks a lot for sharing your insights with us today. Thanks also to the participants to join us. I hope you found the session valuable. I'm pretty sure you have. Now it's time for the Q&A. So please feel feel free to drop your question in the Q&A section, and we will read them out for a speaker to answer. So let's see, we have 2 questions already. So we have one question from Kerry. Thanks, Kerry, for your question. Can peer-reviewed literature be used to justify non-clinical studies required by manufacturers with real patient samples? And then she has modified the question and she has said, for established devices from the IVDD. I'm not sure if it's clear enough, Jordan Clemens.
Yes, it's clear. Thank you.
Okay.
So the answer is yes. If you have peer-reviewed based on your device, and as we mentioned in the presentation, you may be sure that the clinical performances that have been made on the same version of your device. That there is no change in intended use, there is no change in raw material, etc., etc. All the points we mentioned. If it's the case and you have sufficient data from peer-reviewed literature, you don't have to do another clinical performance study.
Okay.
I hope it helps.
Yeah. We have another question from Sonia. Thanks, Sonia, for your question. How can a Class C device being certified under the IVDR without clinical data, no in between gaps, no data shown in the IFU and SSP, and no hint to MPF, sorry. Not sure if it's clear.
No hint to PMPF, no data shown in UNSSP. Okay, this is a very specific question. It depends a lot of what is your intended use, what your device is made for, because just with that we cannot make an answer.
Yeah. No worries, Jordan. It's okay. Sonia, I invite you to ask this question by email. As Jordan said, maybe it's a bit specific, so we can address this by email, and I'm pretty sure you will have a proper answer. So maybe we can move to the next one. Julian is asking, scientific validity can be partly demonstrated with a proof of concept study.
Yes. Yes, partly, you said the word. There was a slide where all sources that can be used to demonstrate scientific validity are specified in the IVDR. So a proof of concept study, yes, it's basically a good point to start for scientific validity, because you may prove the link between your analyte and the clinical condition you want to diagnose. So yes.
Okay, good. Let's move. Aurélie has another question. What are the constraints for a healthcare establishment for an IHC test, for example, in terms of inclusion criteria?
I'm not sure to understand well the question. In terms of inclusion criteria, I think it depends of the target population. Maybe you can reformulate your question already, or send it by email. We can go further on it. Just with that, sorry, I don't know what to say.
We can reply to you by email, Aurélie. We can definitely address this question by email. This is maybe a bit specific. We can do that. Okay. So we have another question for Damien. Can a validation work that was not initially included in the performance evaluation plan be subsequently included in the clinical performance evaluation report?
Okay. Yes and no. Because in your performance evaluation plan, you have to present the method you will use to demonstrate scientific validity, analytical performance, clinical performance. And you don't have to detail all the tests you will do. So yes, you can add, I don't know the term you use, but you can add a study or a test you made later in the performance evaluation report, but you have to detail all you have to add in annex the protocol of the test you made and of course the report of this test. So yes, you could add several other tests you made.
Great. Thanks, Jordan. Okay, we have another question from Irene. Thank you very much for this webinar. Thanks you for attending, Irene. How would you face the analytical validation for a software IVD?
Yes, good points, because software are different from a classic IVD, and we have the same problem in the medical device. I invite you to read. I think you already read it, but the MDCG 2020/1, they present in detail all these points: scientific validity, the analytical performance, the clinical performance for software, IVD and medical device. And there are several examples at the end of the document that can help you. But analytical performance for a software, you may have specific analytical criteria that are different from a classic IVD. But you also have the classical ones such as sensitivity, analytical sensitivity, analytical specificity, etc. for your software. So yes, it's very clear in the MDCG guide.
And if I may, Irene, I invite you also to send us an email to continue the discussion on software and specificity for software. We have at Strategical and PLG expert on software regarding CE mark.
Yeah, that's true. As Clemence mentioned, we have a really good team specifically for software of medical device. So Irene, I invite you to email us, and even we can have a quick chat further days. Okay, so let's move to the next question. Hela had another question. Are there categories of IVDs that are particularly at risk of being removed from the market due to performance data gaps? Sorry.
Yes, I think it depends of the class risk of the device. I think it's obvious that a higher class risk may have a higher risk to be removed if you don't have if you don't have any or sufficient data. It's a good point, because we know that with a regulation, a lot of manufacturers that before in the directive in auto certification may have to do a technical documentation and have a notified body evaluation. So it's a good point to ask if we have sufficient data. And of course, as I said, higher class risk of medical device have certainly more risk to be removed from the market.
Okay. So I see that we have further question from Kerry and Damien that previously asked. As I see that they are a bit more specific, I invite you, Kerry and Damien, to answer this question by email. We will have our team here that probably will answer this question better in a specific email. But I don't see any other further question. So I think that, yeah, we are done. I think that we can close the webinar. So, all right, let's close this. If your question wasn't answered, don't worry. We will, as I previously mentioned, we'll make sure to follow up with an email. Thanks again for joining us today. If you have any further question about our services for IVD and even for software, as you were mentioning, feel free to reach out to me directly. I will be happy to answer you, to present those services. Just a quick reminder, we will have another webinar tomorrow at 11 also. It will be focused on the AI regulation, which is a really hot topic. So I invite you to be there. We would love to see you there. And also don't forget to follow PLG and Strategical on LinkedIn to stay updated on our upcoming events, webinars, and everything. So have a great day and see you soon. We can close the webinar.
Thank you very
Goodbye.
much.
Thank you.